The final review. So we are going to do a focus on the most new material since exam 2, which is going to cover calcium, phosphorus, and then its closely related regulatory vitamin D. But you can't talk about vitamin D without talking about vitamin K because they also go hand in hand. And then of course we've got the fluid dynamics with the electrolytes, because the electrolytes of the macro minerals very tightly are coordinated with the fluid balance. And then you can't talk about fluid balance and the electrolytes without talking about acid base and control of acidity. And then there's a very heavy focus on very key concepts for cumulative that are important that we carry through. So we'll take a quick look over at those as well. The final review material has been updated on Signet. The prior semesters was uploaded. I do apologize. That was an oversight on my part. I uploaded the wrong final review. I do apologize. That the Word document was wrong. However, I had posted, and I hope you paid more attention to the weekly material, that the new material was bulk of the exams. So I hope you put a heavy emphasis into the new material and less so on the older document. Since nobody really emailed me that there was a question except one, and it was a very indirect question, so I apologize. I'm hoping that the majority of the class has not looked at this yet, which is why you did not realize the discrepancy. So hopefully by Wednesday, when you have your exam, you will realize that you can now study off of the correct information. And good luck on the other finals, and hope your practicals went good. So let's jump right into calcium and phosphorus. So appreciating that calcium is one of the most abundant macro minerals is important, and it has very important physiological roles. And calcium and phosphorus go hand in hand because of that hydroxyapatite matrix, which is that calcium and organic phosphate. So phosphorus in the body pretty much is in the organic PO43 minus ionic state or organic state, and most of it is going to be with calcium in teeth and bones. And then of course the hormonal regulation for tight regulation of serum calcium also influences the phosphorus levels as well. So 99% of calcium in bones is as the hydroxyapatite, and of course bone demineralization is going to be through PTH and then vitamin D, whereas bone remineralization is through a vitamin K dependent reaction, which we look at in a few. In addition, of course, to teeth and bone as hydroxyapatite, calcium is also really important in the other roles of calcium here is going to be understanding that in addition to serum calcium and bone and teeth calcium as hydroxyapatite, calcium is a really potent second messenger, and that means that it's going to have very regulated mechanisms to kick calcium out. So calcium has to be, it stimulates the cell, and then it has a direct response, and then the cell immediately kicks it out. Because if you have too much calcium in the cell and if you have too much calcium in the serum, it will continue to have its active roles, and it can be overstimulatory and aggravate some of the contractions and nerve impulses, and then it'll continue to stimulate. So we definitely have a tight regulation of calcium, both in serum, which we learn hormonally and we'll review in a moment, but also intracellularly. As soon as calcium does its job intracellularly, that cell is like, buddy, get out, and it pumps it right back out. So that's important. Okay, so calcium is the most abundant, found in what two primary places? Teeth and bone, and it's hydroxyapatite. Right, so what four molecules regulate blood serum? And of course, that is going to be your two hormones that are going to respond to a drop in serum calcium, and that's going to be your PTH and your vitamin D in its active form, which is the calcitriol. And then the other two, which are the antagonists to PTH and calcitriol, which is your active vitamin D, which are going to respond to elevated blood calcium, is going to be your thyroid secreted calcitonin and your osteoblast secreted fibroblast growth factor 23, FGF23. So I did go ahead and post an update. So this is on Signet, and I put it under week 12 to go along with the calcium from that week. So here is a nice description of the feedback loop, and you do have to appreciate all of this. So I'm going to, which one decreases, which one elevates. So we're going to cover this in just a second. Okay, so the active form of vitamin D, of course, is your calcitriol, and what opposes PTH, of course, that's going to be both your calcitonin and your fibroblast growth factor. What 3 locations does the PTH act on? That is going to be the kidney. It increases resorption of calcium while increasing the rate of excretion of the phosphorus. So it saves calcium and gets rid of phosphorus. PTH also acts on vitamin D at the kidney. And then PTH is, of course, going to act on the bone resorption along with vitamin D. That would technically be 2 locations. Then your vitamin D, your active calcitriol, is going to act at your intestines. So, okay, so vitamin D acts in 3 places, PTH acts in 2 places. Vitamin D is going to increase the absorption of intestinal calcium. It also works with PTH at the level of the kidney, and then also works with PTH at the level of the bone, okay? Which one responds to elevated phosphorus? So your fibroblast growth factor is the one that's going to respond to an elevation in phosphorus, and then calcitonin responds to an elevation in calcium. Now, I know this can be confusing. So on this review, I had already updated this. So the calcitriol, which is your active vitamin D, increases the gut absorption when the vitamin D and the PTH are active. Then once the feedback loop increases your serum calcium, then the antagonists kick in and they act to inhibit calcitriol and PTH and the gland and the parathyroid gland, while also decreasing the gut absorption because we don't want calcium to rise. The signaling roles are what I originally looked at, so I'm going to stop there for a second and then pop back to Signet. Okay, so you have to appreciate that calcium regulation is a feedback loop. It's a stimulus, okay? So the calcium serum level drops, and then that's a stimulus or a signal that that the hormonal regulatory organs need to kick in to secrete the hormones that will prevent further drop. So low serum calcium, because remember, it's under tight hormonal control. Low calcium serum and plasma calcium is going to first stimulate the parathyroid gland to secrete the PTH, the parathyroid hormone. That travels from the parathyroid glands, which are sitting on your thyroid. There's 4 little nodules, and they travel in the blood to the kidneys. Once they are in the kidneys, they activate that renal 25 vitamin D into active 125 calcitriol. While PTH is also in the kidney, it also affects the resorption at the level of the nephrons, as we had said, to increase the saving calcium, so resorption, while increasing the loss of phosphorus. So the PTH is going to act on the bone as well to resorption, degradation, demineralization of the calcium phosphorus hydroxyapatite. So the active vitamin D calcitriol, once it's activated from the PTH from the kidneys, acts on the 3 places we had said is going to be the intestines to increase the absorption of dietary calcium, to also it works with the PTH a little bit at the level of the nephron, and then primarily also with PTH at the level of bone resorption to drop the calcium and the phosphorus that is degraded out of the hydroxyapatite matrix into blood serum. So the blood serum is going to have an elevation of both calcium and phosphorus, because bone resorption is going to be the hydroxyapatite matrix, and both are being secreted by bone demineralization into serum. And then the gut resorption is bringing them in, but then the PTH at the nephrons is going to keep the calcium, but then try to get rid of and balance some of the phosphorus. So as serum calcium rises and as serum phosphorus rises, now they reach a point where once their levels are high enough, then they stimulate the organs that are the hormonal regulation, which is the negative feedback loop, to secrete the antagonist to halt the process of demineralization and to deactivate the PTH and the vitamin D calcitriol. You do not want excessive calcium in the blood because it's a second messenger. It can aggravate the heart muscle, it can aggravate the smooth muscle, and it can overstimulate the tissues. Remember, a tight regulation is for a purpose, and the body designed it that way. So once calcium rises, it stimulates calcitonin to be secreted by the thyroid gland, and then that's going to inhibit the parathyroid from secreting more PTH. It also helps deactivate some of the vitamin D. And the second response is as the phosphorus rises, because that happens simultaneously, the backup method is the osteoblast secreted FGF23. So it responds to a rise in serum phosphorus to also inhibit and stop the parathyroid gland from secreting PTH. It deactivates the PTH, and it also acts at the kidneys to stop absorbing and activating vitamin D. So it's an antagonist to the PTH and the vitamin D. And then the calcitonin also it antagonizes the intestinal absorption, and it halts and decreases that upregulated calcium absorption. So that is how we prevent a too much rise and a too much drop. So that whole thing is the feedback loop. So first it rises, you have the hormonal responses, and then in return, you then have to turn that off so that you don't have excessive secretion and demineralization. Okay, so that should bring us up to here. So know the different places that phosphorus is going to be important in its physiological roles. So not only is it participating with calcium in the hydroxyapatite, we also have it as an intracellular. That's one of our negative buffers. It's an intracellular buffer, which we will review in our acid base in a few more bullet points for review. It's a very important intracellular anion, which is the acid buffer. And then, of course, it's going to have a structural role in several different types of tissues. The phospholipids is primarily a major source, hence phosphorus, phospholipid. So it's a phosphorus on a lipid head. And then you have your RNA and DNA, so it's part of the backbone. And then very important in the red blood cell is the 2,3-DPG, and that is bound with hemoglobin, and that's important for delivery of oxygen in the release of oxygen at the tissues. And then we also have the high phosphate bond, just like your ATP. And then when ATP does its job, then it loses that high phosphate bond and becomes ADP, adenosine triphosphate. So it also is that high energy bond. So know the different functional roles of phosphorus. The demineralization is going to be as we had said, PTH and calcitriol, and it releases both calcium and phosphorus into the serum because of the hydroxyapatite being degraded. And then remineralization, of course, is going to be a vitamin K dependent carboxylation reaction. So the remineralization of vitamin K through the osteocalcin is a carboxylation reaction. The carboxylation reaction is with a vitamin K dependent cofactor redox role that the GLA protein requires in order to help get calcium out of the serum and put it into the bone matrix to allow for remineralization back into the hydroxyapatite. So the vitamin K dependent osteocalcin is a carboxylation, and that's part of bone remineralization. The vegetable that is high in the vitamin D, of course, is going to be the shiitake mushroom. And as we look at the vitamin D as well, the D2 is less commonly supplemented. We normally have a D3. The cytokine that is important for that vitamin D interacts with, of course, is going to be the Tregs, and vitamin D increases the interleukin 10, and the interleukin 10 is going to be a very important regulatory Treg that helps prevent against autoimmune and keeps the populations of T1 and T2 in balance. When you lose the balance, you are more prone to autoimmune attacks. So having a healthy interleukin 10 is really important. And we review some of that again next semester in GIGU because that comes into play in the gut-associated lymphoid tissue. Let's see, what fat-soluble vitamin does active vitamin D in the VDR function and require? The VDR is the vitamin D receptor, and this is one of the, it's the non-vitamin physiological role that vitamin D has, and it's at the genetic level. So vitamin D not only has the immune and the bone and calcium homeostasis functional roles, but we have learned that it's very important in epigenetic control at the level of the VDR. And the VDR is the, again, vitamin D receptor, works at the transcription level of the genome to turn on different vitamin D receptors. However, it works with vitamin A because it has to be part of the retinoid RXR heterodimer. So not only do you need sufficient amounts of vitamin D, but in order to have the VDR, vitamin D receptor, at the level of epigenetics properly functioning, you also need vitamin A. K1 is phylloquinone and K2 is menaquinone. And this is important because the phylloquinone primarily works as K1 on the clotting cascade, whereas K2, the menaquinone, is going to be more important for the other vitamin K dependent carboxylation reactions at the level of preventing arteriosclerosis and preventing osteoporosis. So K1, phylloquinone, works at the clotting cascade. And then the GLA carboxylation reaction kind of works in opposite directions, and it has to do with binding a calcium. And then for bone, the carboxylation, which is the vitamin K dependent reaction, takes calcium out of the blood and puts it into the bone matrix to allow for hydroxyapatite remineralization. Whereas the vitamin K dependent carboxylation reaction in the blood vessels kicks calcium out of the blood vessel, and it actually prevents the arteriosclerosis because the calcium will form hydroxyapatite crystal with phosphorus, just like we do for bone, and that's basically the arteriosclerosis process, and the vitamin K prevents that from happening. So the menaquinone supplementation, MK4 and MK7, are really potent vitamin K supplements that we can take with vitamin D. MK7 tends to have a little bit of a longer half-life. It sticks around longer. It's a little bit more potent. Vitamin K2 is what we just said for arteriosclerosis. All right, this is super important. So warfarin and Coumadin are the primary medications that are your blood thinners. And they have come to realize that because of the reaction where it inhibits the functional redox recycling of vitamin K. So just like our antioxidant reducing agents that gains and loses that hydrogen atom, vitamin K does too. So through a redox reaction, vitamin K becomes active, then it's inactive. So it's active when it has its in its reduced form that allows that carboxylation reaction, which is the physiological function of the vitamin K. And then through that carboxylation reaction, it becomes oxidized, and then it has to get reduced back to gaining that hydrogen atom in order to do another round of carboxylation. So the reason that warfarin and Coumadin are blood thinners, because knowing that the vitamin K is important for that clotting cascade, warfarin inhibits the ability for vitamin K to get recycled. It keeps vitamin K in the oxidized, inactive form. So the three things are that not only does warfarin and Coumadin are drugs, medications, however, baby aspirin, which is a salicylic acid, also mimics the drugs. They're doing the same things, and they inhibit vitamin K's recycling. Now, we can use white willow in doses of 750 milligrams or more. This is a wonderful anti-inflammatory natural agent. It's a naturally occurring salicylic acid, whereas a salicylate. Aspirin is a synthesized, synthetic, fake test tube made salicylic acid. However, white willow in all of its components has many anti-inflammatory properties, but at doses of 750 milligrams and greater, it actually will mimic a baby aspirin. That's really great for somebody that we want to get off of baby aspirins, because you know that it'll eventually destroy your gut lining. It can cause an intestinal bleed. So it's a great substitution. However, too many people incorrectly take baby aspirins and they don't realize that they are inhibiting their vitamin K. So even a white willow dose, which is a natural agent, it's a natural salicylate, it also will inhibit the vitamin K recycling, which means you're inducing a vitamin K deficiency. So if you take white willow at 300 milligrams in the morning and 300 milligrams at night, you don't have to worry about it. It has to be that high dose that mimics it. So I want you to know all three of these, and you need to know the dosage. 715 milligrams and above is what the dosage is that inhibits vitamin K. And the vitamin K must be recycled, and that's what we were just talking about, is the physiological role of the vitamin K is going to be the ability to do that carboxylation reaction. And the remineralization, you got to recognize, is that osteocalcin, which is the vitamin K dependent carboxylation reaction that takes calcium out of the blood. All right, moving into fluid dynamics and acid base. The minerals, of course, that are going to be your electrolytes are the ones that are involved in working with water to sustain blood volume. And then, of course, different hormonal regulations will then sustain and maintain blood pressure. So the three macro minerals that work for fluid balance and ionic movement are going to be our sodium, chloride, and our potassium. The electrolytes work together to, I just said that, with the fluid balance, with blood volume, and then of course downstream from that is the physiological blood pressure. The important thing about the compartments is that it's going to be how the acid base as well as the solute is maintained. So you have intracellular and you have extracellular compartments. Extracellular fluids, but then your extracellular is made up of the capillary plasma. The capillary is a permeable membrane that abuts the tissue fluid when you're in the capillaries, and the capillaries travel, being the smallest, that's where gaseous exchange is happening. Then that's your interstitial fluid, which is the fluid that surrounds and bathes the cells as an organs. So your extracellular fluid is a combination and summation of your plasma plus the tissue interstitial. And these guys, these two these two fluids are in equilibrium together. So water can freely pass between the capillary, and that as well as some of the ions. Now, we have albumin in the plasma in the blood vessel to prevent loss of water. That's your oncotic pressure. So that does not go through your capillary permeability, nor does your red blood cells or white blood cells, unless you've got a problem with edema or you have damage. So in a normal healthy blood cell, you can exchange ions and you can exchange fluid. Now, that means that we are in equilibrium. So if your tissues are acidic, so is your blood. And we have to work really hard at the acidity. And we'll talk about that in a second. Now, the difference with the intracellular, so now we have our cell membrane. So the ions are different. So our primary cation, positive extracellular, is sodium. And then our positive cation intracellular is potassium. And that's important because of the sodium-potassium ATPase pump. We spend out, I think it's almost like 15 to 20% of our basal metabolic ATP generated, or BMR, to simply sustain the sodium-potassium ATP pump, the ATPase, because if we lose this, the cell will swell, and then we will also lose the ability of our action potential, the voltage that we require across the different membranes. So we have fluid freely exchangeable, and when we have dehydration, we lose water from inside, intracellular, to the extracellular fluids, and basically we can cause like a little bit of shriveling. So hydration is important, not just for blood volume, but also to make sure that we have enough fluid inside the cells itself, so that we don't disrupt enzyme function and transport of ions and proteins and enzymes such in and out of the cell. So the intracellular fluid can exchange with the extracellular fluid. However, the ions do not freely pass. There are channels and there are transmembrane carriers. So in order for things to get inside and out, there is actually a very specific, protein or again, channel or carrier, which is different than the solute, which can freely travel between the extracellular compartments, okay? So plasma consists of the non-fluid portion of blood and the non-cellular. So plasma, excuse me, it's the opposite. Plasma is the fluid content that has, that is not made up of cells. It's not white blood cells and it's not red blood cells. It's the fluid and everything else. Which electrolytes are inside and outside, and we already talked about that, the ATPase. Which solutes are found in each. So in addition to sodium being in the extracellular, the other primary one is going to be our chloride and our bicarbonate. Bicarbonate is a very important anion in blood. It acts as a very important buffer, and it's also important in the acid base, which I'll explain in a moment. And then our organic phosphate with potassium is going to be primarily our intracellular, along with magnesium and sulfate. Can the electrolytes freely pass? As we just explained, no, there has to be channels or protein carriers or very, very specific systems. They intracellular do not, but water does. The organ that primarily manages fluid balance and blood volume, of course, is going to be through the kidneys, and that's going to be through the nephron resorption and or secretion mechanisms that manage the different macrominerals and electrolyte and fluid balances. How much blood becomes the filtrate? So this is important as well. So when we are looking at the renal function, of the nephron. The nephron, of course, is what makes up the kidney's structural unit. And Dr. Ott's class and for boards, you need to know what's absorbed where in the loop of Henle and proximal and distal convoluted tubules. But for us, you need to get the gist when we're looking at how it functions as a whole. So you have 22% of the cardiac output going through the renal arteries, and then that's going to be what forms the filtrate. The filtrate goes in through the glomeruli and becomes the filtrate. Then through the renal nephron structures at the different control points, we resorb nutrients, amino acids, and glucose, and then majority of our electrolytes and macrominerals and different minerals through nephron regulated resorption. What we don't resorb actively stays in filtrate and becomes urine. It's a really great way for us to control rapid elimination of metabolic waste and any conjugated toxins coming from the liver that have been marked for excretion, because they rapidly go from that cardiac output through that renal artery right into filtrate, and then they go right into urine. And there's no resorption. We don't take them actively back up into blood like we do with the different minerals and nutrients, our glucose and our and amino acids. Okay, so the difference with resorption versus secretion. So tubular reabsorption is what is returned to plasma through that active nephron control points that takes them out, pushes them, or transports them out of the filtrate and puts them back into blood. The difference with secretion is the substances that are in blood that did not enter the filtrate, they can be actively, purposely pushed into filtrate as a control point to allow excretion if we need to regulate what's in our serum. It's one way that we tightly control that we push intentionally basically into urine to eliminate. It's a forced excretion that we want to control if it didn't, the 22% that goes into filtrate. How does the filtrate become or not become urine? We just said that. If we don't have an active nephron resorption mechanism, then it remains in filtrate, which will then ultimately become your urine. If there's a reabsorption mechanism because of the nephron and hormonal regulation or a transport, then it's taken out of filtrate and put back into blood, and then it does not become urine. So I had just said this as well, because of the 22% of cardiac output from the renal artery that forms filtrate, that is a rapid way to get rid of metabolic waste and toxins, and then there are no mechanisms to reabsorb them, so we put them right into urine. And then what hormone controls water? Okay, so the interesting thing is we do not reabsorb either potassium or chloride. So we do reabsorb the sodium and the water, and there are different systems for reabsorption. So when we have a drop in serum water, a drop in blood volume, we stimulate the renin-RAS aldosterone system. The end result of RAS is the adrenal secretion of aldosterone. Even though the stimulus is a drop in fluid, a drop in blood volume, the action that happens at the level of the kidneys is aldosterone increases the rate of sodium reabsorption. When we increase the amount of salt, water follows, and then we can raise blood volume, the amount of fluid in the blood vessels. So that causes sodium retention and water follows. ADH, which is also called vasopressin, comes from the posterior pituitary. ADH is anti-diuretic, meaning we're stopping the loss of water. And this is going to tell the kidneys to increase the aquaporins, and we save water. alcohol inhibits ADH, which is why we can become dehydrated with alcohol, because we are not saving our water through the aquaporins because the ADH has been inhibited in the posterior pituitary. And then the natriuretic peptide is it opposes and is an antagonist to the aldosterone. So instead of saving sodium, the natriuretic peptide will actually increase its excretion so that we urinate out sodium. So if we have a potassium sparing diuretic, it doesn't resorb potassium. It just doesn't cause the excessive loss, because under aldosterone, there's a dual effect. Aldosterone is going to save sodium, but we're going to increase the rate of loss of potassium. And that's a problem because we are already backwards in our ratios of potassium and sodium because of our Western diets and decreased ingestion of our produce. And the primary sources of potassium dietary are going to be from fruits and vegetables, and processed foods are very there's virtually like no potassium in them. It's all salt. So when we have aldosterone, we save sodium but lose potassium. So then the potassium sparing diuretic just makes sure that we don't lose extra potassium, but there's no mechanism to resorb it. We do not have a mechanism for chloride. However, in fluid dynamics, similar to water, chloride is going to, because of the ionic charge, is going to very commonly follow sodium's physiodynamics. All right, so acid-base. Acid-base is so important because as we regulate the hydrogen atom, we are going to never save it. We do not preserve any acids. There are no mechanisms. All of it gets excreted, and then inside the tissue, outside the tissue, at the level of the nephron, at the level of the lungs, and inside the blood, which basically covers the entire body, we have mechanisms to mop up the acids. We do not want them in excess because that would be acidity. We also have to be careful to not be too alkaline for too long. It's good to alkalize, but we don't want to be in alkalosis, which is long-term too alkaline, not enough of the acids. So our body's pH is tightly regulated at that at the 7.2, 7.3 to a 7.45. So we need to physiologically stay in this range. However, many people run on the acidic side, and that's because if you're not actively alkalizing with minerals and fruits and vegetables and produce, and that's where your potassium comes from, and the way that the body preserves alkalinity is lost in our Western diets. So it's very important that we supplement with minerals, because the minerals are going to act to help mop up some of those, some of the acids. So we have the chloride shift, which happens in the red blood cell. Let's see where my picture is. It's got to be down here somewhere. There we go. So we have the chloride shift, which is going to work with the level of the bicarbonate. And bicarbonate is important because bicarbonate is one of the best buffers that we can have. And citrate is an excellent anion that we can dietarily use. So I'm pretty sure. So citrate is the dietary anion, and it's going to be an excellent acid buffer, just like the organic phosphate is the intracellular anion that mops up the hydrogen atoms, cation. Then our bicarbonate is the buffer in the blood as well as the red blood cell. In addition, the red blood cell also has the hemoglobin, because proteins are also a good, because of their charge, they can help to accept or release the proton as needed, the hydrogen atom. So metabolic waste at the level of the tissue is going to include that CO2, that carbon dioxide. So we take oxygen, we oxidize it through the electron transport chain to get our ATP for energy, but the metabolic waste product is that CO2. CO2 diffuses through the tissue interstitial fluid into the plasma, and then it rapidly disassociates in a reversible direction with that zinc-dependent carbonic anhydrase, and the CO2 rapidly becomes bicarbonate. So then bicarbonate can be inside the red blood cell as a buffer, or it can do that chloride shift so that we can kick the bicarbonate out, and then bicarbonate will be in the serum to act as a buffer in the blood, in the plasma. So then the hemoglobin itself can mop up the acids, and then the the bicarbonate as well. So bicarbonate and hemoglobin work together as the buffers to mop up the acids. And then there's a really cool mechanism at the level of the kidney as well, where the exchange, the kidney is one of the top regulators of acidity, and it does so by doing different ionic exchanges. So bicarbonate, organic phosphate, ammonia, and glutamine are wonderful. Glutamine can kick off, glutamine has 2 of the amino groups, and you can kick off one, and that amino group can then mop, which is NH2, NH3, and then it can scoop up and become an ammonium by grabbing an acid, and then we can urinate that out. So glutamine is an excellent acid-base buffer as well. So you can supplement with glutamine as an acid base. It also is really great for your gut. If you remember, it's the one of the top fuel sources that the enterocytes use. Let's see. So the Western diets, as we had said, are not only are they backwards, the Western diets include high salt processed foods, and then in addition to that, the people are not eating the produce and the fruits and the vegetables where the primary dietary potassium is coming from, and then basically the other food groups, which are just bland, they're just void of vitamin K. So vitamin K is really important to sustain. I didn't mean vitamin K. I saw the K. I meant potassium. I'm sorry. I meant potassium. So the K plus is potassium. The vitamin K is your menaquinone and your phylloquinone. All right, so the cumulative is going to, these are statements, not so much questions, right? We wanted to make sure that we remember that the GLUT4 translocation at the skeletal muscle and adipocyte, which is that insulin-dependent reaction where insulin binds, that's insulin sensitivity, and we want to improve the translocation of the GLUT4 through the activation of the AMPK, and we do that with chromium and vanadium. Exercise itself also stimulates the activity of the GLUT4 translocation to support insulin sensitivity. Chromate is it's the best supplemental form that is a niacin-bound chromium, because chromium with valence of 4 is the most bioavailable, and that's also the form that the tissue uses in the insulin receptor complex. So chromate is the best. A selenoprotein is a protein bound with the selenium. So the selenium bound to a protein, especially a methionine, so you see a selenomethionine, that's a more bioavailable way to supplement for selenium to increase its absorption as well as its bioavailability. Selenium is important as the antioxidant because of the selenium-dependent glutathione peroxidase. Remember, that requires 2 glutathiones, and that is a selenium-dependent intrinsic antioxidant. And then the thyroid is going to be a selenium-dependent deiodinase, and that's important because the T4 thyroid hormone is higher quantities in blood. However, T3 is more active, and the selenium-dependent deiodinase, removing iodine, an iodine, that's how you go from a T4 to a T3. So at the peripheral tissue, the tissue needs sufficient amounts of selenium deiodinase to convert T4 into peripheral T3 to have an active form of the thyroid. So selenium is important for that. Heme is required, and remember, heme has many, many, many functions. Heme is going to be your succinyl-CoA plus your iron, and that is your iron porphyrin molecule. So tyrosine plus iodine is going to have that heme-dependent reaction that allows for the first synthesis of the thyroid hormone in the thyroglobulin. Iodine is pumped up its gradient, it's ATPase, so iodine has to be pumped up a gradient into the thyroid gland. And then the best ways to supplement for sufficient iodine in the body is Lugol's solution, which is a combination of iodine and iodide. And then kelp is a really wonderful natural seafood source. And then potassium iodide is either a supplement or you can buy it as a salt. Sulfur amino acids, of course, I hope you remember that that's a thiol. And then our sulfur amino acids are going to be methionine, cysteine, and taurine. If you recall from diet and nutrition, methionine catabolizes into cysteine. Cysteine catabolizes is broken down into taurine. And then cysteine plus glutamate and glycine are the tripeptide that synthesizes your glutathione, which is the intrinsic antioxidant that is both an intrinsic antioxidant enzyme system as well as a solo antioxidant. It's the most potent intrinsic antioxidant we have in the body. Sulfur is vital for your liver conjugation. Remember your sulfur foods, your onion, your garlic, your brassica, those are part of the phase 2 liver conjugation detox reactions, because we have to be able to conjugate any chemical, hormone, over-the-counter, anything that's a xenobiotic is going to have to be marked for excretion, and we do that with liver phase 1 and liver phase 2, and sulfur for phase 2 conjugation marks it for elimination and is very important. As I had said, those come from the sulfur vegetables, and we want that very potent sulfur antioxidant that's in the brassica sulfur vegetables, which is that sulforaphane. Molybdenum is important because it's part of sulfur metabolism, and it's also a important cofactor that is a redox agent. So it increases the sulfur bioavailability. It participates in xanthine oxidase. However, it's that multi-mineral redox reaction. Nuclear factor kappa B, of course, is your pro-inflammatory transcription factor, increasing the rate of inflammatory cytokines and inflammation. Your NRF2 is that antioxidant and transcription factor that increases the synthesis of your antioxidant enzymes, as well as increases the rate of synthesis and bioavailability of your phase 2 conjugation enzymes. Super important. Copper and zinc is part of your superoxide dismutase, and that's the intrinsic antioxidant system enzyme that is in the cytosol, and that takes superoxide dismutase and converts it to hydrogen peroxide. The same superoxide dismutase, but has a different cofactor mineral, which is your manganese, which is inside the mitochondria, and of course that does the same thing. It takes your superoxide dismutase, which is always being generated at the electron transport chain, why this guy is so important, and it converts it then to the hydrogen peroxide. But remember, we have 2 more intrinsic antioxidants, because hydrogen peroxide is still a potent free radical oxidizing agent. So then we have our selenium-dependent glutathione peroxidase, and that converts your hydrogen peroxide to water. And then, of course, you also have your heme-dependent catalase, which also, alongside your selenium-dependent glutathione peroxidase, is going to convert that hydrogen peroxide to water as an intrinsic antioxidant. Know that CoQ10 is in 3 of the 4 electron transport chains, and then the copper iron is part of the electron cytochrome C, but then we also have heme-dependent complex with sulfur also in the electron transport chain. So copper, iron, right, B2, B3, and I just said iron, copper, iron, oh, and sulfur, all super important. So, I mean, that's a handful of minerals and vitamins already that and CoQ10, just to power the electron transport chain. Don't forget your succinyl-CoA. Zinc is your carbonic anhydrase, which we talked about just a short while ago. It also is important in the liver for alcohol dehydrogenase. It requires 4 zincs, or actually 6 zincs to detox alcohol. And then zinc is also important at the level of gene transcription and modulation through zinc fingers. Copper is transported on the ceruloplasmin, and don't forget that ceruloplasmin, definitely a board's question, is going to participate in getting iron to go through the redox state that is required in order to allow iron to get onto transferrin to be transported in the blood. Super important. Iron as heme is a catalase, which we just said. It's also important for the synthesis of your neurotransmitters. And we also mentioned that heme is also important for the synthesis of the thyroid hormone. And heme is part of your catalase. So that's mood and anxiety, part of your neurotransmitters. That's also anemia because of hemoglobin. Then you've got the protection against oxidative stress, which is because of the heme catalase. Then you've got thyroid protection because of the heme acting with the thyroid hormone synthesis. And those are just a few examples that I really want you to remember of how important heme is. So I hope I've pounded this into your brains. Heme requires your succinyl-CoA plus glycine. That's the first step that synthesizes it, to synthesize heme. It's about 8 or 9 steps. And your succinyl-CoA, just to review, because you're going to have to be tested on this in diet and nutrition anyways, right? Your 2 branch chains, isoleucine and valine, along with methionine and threonine. All 4 of those amino acids are going to be completely catabolized to a final step that is going to synthesize the succinyl-CoA, and all 4 of them require that adenosyl B12 as the final catabolic step to get to succinyl-CoA. Symptoms of low iron, that's what I just said a minute ago. Fatigue, slow healing, mood, anxiety, and depression. Fatigue is basically because of the hemoglobin as well as the oxidative stress. You're more prone to oxidative stress and slow wound healing if you have an inefficient electron transport chain because it's heme dependent. Anxiety, mood, and depression because of thyroid hormone as well as neurotransmitter synthesis, both being heme dependent cofactors and the enzymes that are synthesizing them. So understanding, carrying this forward is so important, that iron heme is not just anemia. It's so important for other physiological roles. And heme is a very potent, vital cofactor for many enzymes. Dietary heme is absorbed as heme. So people who eat meat and eat blood, flesh, carnivores, are going to have a better absorption because the body does not have to do any change to the heme. You have to have sufficient digestive enzymes in order to break out of the heme, out of the food matrix. However, the intestinal brush border absorbs heme as heme. And the difference with dietary non-heme is it has to go through that redox reaction, which is why non-heme plant-based iron is best eaten with vitamin C, because vitamin C is going to participate in that redox reaction to allow for the uptake of the redox changed heme iron at the level of the enterocyte of the brush border. So that is a great review of some of the key concepts that we learned throughout this semester, and then a nice heavy focus on the last material. I wish you the best of luck, and I will see you next semester in GIGU.